The fourth reason I will talk about is K2 and its interaction with the microbiome. Remember to please like and subscribe if you like this type of content. But what is the microbiome, you might ask? Well, think of it as something that has these little microscopic players. You have these microscopic players all within the outside of your surfaces, the throat, the stomach, the intestines, and outside your skin. But isn't the digestive tract inside your body, you might ask? While that is true, it also connects to the outside. Think of it as a tube. Since the tube connects to the outside, these little players can spread all throughout the inside of the tube since it connects outside. In fact, these little players outnumber your human cells 10 to 1. This means for every human cell, there are 10 of these little players. That's what I mean here, you are more bacterial than human. I'm making a simple comparison here. I'm comparing the bacteria and the microbiome to players, but you are more bacterial than human. Part of this microbiome contains bacteria, and there are 10, for every human cell, there are 10 of these bacterial cells. In fact, you might be more bacterial than human. And not even in fact, you definitely are more bacterial than human. So let's look at this more in detail. So this is the anatomy of the human digestive tract, and this is the microbiota that can be found there. Remember that the digestive tract is like a tube, and it is connected outside, and the inside of this tube has these little players. Now, what are these little players? Well, you can see over here that the composition of these little players is different on every surface because the environment there is different. So, for example, you have archaea and bacteria. You can think of these as just as bacteria. They're very similar. You also have viruses and you also have parasites. And so if you also see here, the composition of them at every surface is also different. So for example, if you look at the mouth, there's a lot of oxygen in there because that's the type of environment there. So the microbiota that can be found there are ones that are able to utilize oxygen for energy. So, but then if you look at in the intestines, for example, there's barely any oxygen there. So the microbiota or the bacteria that live there, they have to be able to get energy by not using oxygen, also called anaerobic. So another thing is that this diagram is really good for showing basically the different types of microbiota that can be found. But what I just don't like about this is that it implies that, for example, your general microbiome is all fungi, but it is not fungi. Rather, it is a different composition of microbiome that can be found at different surfaces. For example, the urogenital microbiome can be 10% fungi, 20% bacteria, 30% viruses, 20% parasites, and 20% other, or something of that nature. I'm not even sure if it equals 100, but it was just an example. So now let's look at the anatomy drawing over here. As you see, food goes from your mouth into your stomach, and then it goes into your small intestine, and then it goes into your large intestine, and then out through your urine. And as you can see, it is connected to the outside. It's kind of like a tube that goes into your body. Now, remember the ileum here, which is the last part of the small intestine, because this will be important later on. And I mentioned here the urogenital microbiome before. The urogenital it's just meant to reflect the urine and the genitalia. So the urogenital microbiome, digestive tract microbiome, skin, and oral, which is mouth microbiome. So once again, just think of the microbiome at different environments, just to have a different spectrum or a different composition of basically all four of these type that are found there. So now that I've kind of went over what kind of microbiome composition can be found at different surfaces, what are some of the functions of this microbiome? So this really is an area of active research. Researchers are trying to find out basically different things that the microbiome is responsible for. It really has a very high ability to affect our health. So, just some of the things that the changes to the microbiome composition have been linked to are, for example, cardiovascular disease, inflammatory bowel syndrome, development of the immune system, and allergic conditions, metabolic toxins, and autoimmune conditions. So, for example, lupus is an autoimmune condition. So, what are some of the ways that this can impact Alzheimer's disease with the microbiome? So, first of all, in order to, a lot of the studies on the microbiome and Alzheimer's disease are conducted with fecal microbiota transplantation. Fecal microbiota transplantations are just what the name sounds like. They're fecal matter transplantations. But so they basically get the feces from one person or one animal and they transplant it to the gut of another animal or person. For example, in this case, it's using Alzheimer's disease model mice. So they're transplanting it from model mice to model mice. So the thing is, when you do a fecal microbiota or fecal matter transplantation, uh, you're basically there's a composition in the fecal matter and there's a composition in the receiving animal. So the gut microbiome of the receiving animal becomes more similar to the fecal matter transplantation microbiome. So there's a change in microbiome composition because of that fecal matter transplantation. So there are a number of studies in this paper about fecal matter transplantation and its effect on the microbiome. So let's go over the first one. So these are three studies, and in the first study, it's fecal matter transplantation, fecal microbiota transplantation from healthy mice to an Alzheimer's disease model mice. And by doing this, they increase synaptic plasticity, which is the ability of your neurons to make new connections. And it also decreased beta amyloid. So in the second study that these researchers went over, there once again was fecal microbiota transplantation on a different Alzheimer's disease mouse model than the one in the that was said in the first study. But once again, a similar result was seen. It improved cognition and also decreased beta amyloid, but in addition, it also decreased tau protein. Now, I haven't talked much about tau protein, but you can find out more about tau protein in another video, or you can just look it up on the web. And in addition, this also helped to increase immune function, and surprisingly, it also helped to decrease the reactivity of the glial cells, which is a good thing. So, and then in the third study, younger mice had a fecal microbiota transplantation from older Alzheimer's disease model mice. And what this did is it resulted in cognitive decline and a decline in GABA. So before I'll give you some background to understand this. So how our brain works is that the neurotransmitters, there's a balance between excitatory neurotransmitters and inhibitory neurotransmitters. So the excitatory neurotransmitters is glutamate, and the inhibitory neurotransmitters is GABA, or GABA aminobutyric acid. If there's a decrease in an inhibitory neurotransmitter, this means you have less inhibition, which is bad because this results in a cognitive decline. And also, in addition, the GABA was observed. So, additionally, there also was one case report published in this paper, actually involving humans and not mice, which is nice to see. But before I go over this, I'm going to give you some background information in order to understand the study that I'm about to present to you. So, this is the mini mental state examination that doctors give to patients in order to judge their cognitive ability and how they're thinking and see if they're at risk for Alzheimer's disease or if they're in mild, moderate, or severe Alzheimer's disease. So, they ask you a bunch of questions and then they get points. They then tabulate the points and they give you a score out of 30. So if you look at what some of the questions are, they can ask what is the year, season, date, day of the week, month. And if you get all 5 correct, that give you 5 points for this section. And then they ask you all these questions and they get points. Tabulate the points and you get a score out of 30 here. And that's your MMSE score, which is typically a tool to give an objective measure of your cognitive functioning. Now, how do you interpret that score that you get? Well, if you get a score between 24 to 30, this says that you are in the normal range and that your cognitive functioning is at normal. If you get a score between 20 to 23, you have mild cognitive impairment or early stage or middle Alzheimer's disease. If it's 10 to 19, it might be middle stage or moderate Alzheimer's disease. Probably not probably, but it is. And 0 to 9, it's late stage or severe Alzheimer's disease. So let's go over this case study now. So this was an Alzheimer's disease and fecal matter transplantation in humans. So this was an 82-year-old male and he had antibiotic resistant C. difficile infection. So this is an infection which can be treated with antibiotics, but in this case it was resistant to those antibiotics. And so one of the possible treatments is fecal microbiota transplantation, or fecal matter transplantation as I called it. And he received a fecal matter transplantation for a C. difficile infection. And interestingly, a very shocking result was observed. 4 months later, after his fecal microbiota transplantation, his MMSE, as we saw earlier, which was an objective measure of his cognitive functioning, increased from 20 to 29 over a 4-month period. So his score of 20, if we look over here, he was kind of here. He was kind of borderline, because if he just dropped one more point, he would be in moderate Alzheimer's disease. But he was currently in mild Alzheimer's disease. So think of it as mild moderate Alzheimer's disease. And after getting this fecal microbiota transplantation, it increased from this mild moderate Alzheimer's disease to normal. And even on the high end of normal. So that was a very interesting finding. So everything that I've said up to this point, it's been about microbiota and cognitive functioning. And it's very interesting to see the relationship and interaction between the two. But what about vitamin K2 and Alzheimer's disease? Well, in this paper, there are studies that look at this. They don't objectively directly answer the question, but it is very exciting to see a future area of research going in this direction. So first of all, let's look at vitamin K2 absorption in the intestine. And for this, the evidence kind of comes from 3 studies. Now, in this area of vitamin K2 absorption in the intestine, it's kind of debated among researchers right now. Some researchers say, oh, it's not absorbed, and this is the evidence shows it. Other researchers say, oh, it is absorbed, and this is the research that shows it. So what kind of absorption, if any, or the extent of the bacteria involvement is still actively debated in the research community. So I'll go over 3 studies that actually support the absorption of vitamin K2 in the intestine. So the first study is it found that there is more long-chain MK vitamin K2s than MK4 or MK7 located in the small bowel, which small intestine, the small bowel, same thing. So they found a 7.93 versus 0.92. I'm not sure about the units. There were no units in the study, but if I reference the original study, you'll probably find them. In the second study, vitamin K2, so the researchers said this, that the vitamin K2 made in the gut far exceeds human nutritional requirements, and this was speculated by the study. And in the third study, it was an interesting one, and it was in infants. It said that MK7 was absorbed by breastfeeding. So in the breastfeeding milk, there was MK7 present, but MK7 was not absorbed in the formula. Now, I'm not sure if that means that there was MK7 present in the formula and they did not absorb it, or if there was no MK7 present in the formula. But I'm pretty sure that it refers to the first possibility. So, and then they went over the microbiome and K2 a little more specific in these fourth and fifth studies that they were mentioned. So in this fourth study, they used antibiotic treatment in these mice to disturb the microbiome. And what they found is very interesting, that led to lower bone density, lower bone strength, and a lower amount of genes involved in vitamin K2 synthesis. So what this kind of proposes is that in the microbiome, that there are microbiota that actually do produce vitamin K2, and that vitamin K2 is absorbed because one of the functions, as I mentioned in my previous video, is that vitamin K2 helps to carboxylate matrix GL1 alpha protein and also osteocalcin. And what that allows it to do is basically increase the strength of your bones and to mobilize calcium to go to where it's needed to. And that leads to stronger bone density and stronger bone strength. So if there's less amount of genes involved in vitamin K2 synthesis, then the bacteria in your intestine will make less vitamin K2. It will be absorbed. It will be absorbed. There will be less vitamin K2 overall because the bacteria are making less vitamin K2. And overall, this will lead to lower bone density and lower bone strength. That's what this study is suggesting. And in the fifth study, this was also a very interesting finding, is that they used antibiotic treatment, once again, in mice to disturb the microbiome. But this time, the mouse in question also had a low vitamin K diet. So what the researchers found interesting is that this led to gastric bleeding, and the mice eventually died from this. So this is also a very interesting finding. And then there was a sixth study that the researchers mentioned, but this time it was also in humans, which is interesting because most of the studies are in mice and not in people. So this was involving 74 elderly individuals, and the vitamin K2 was measured in their fecal matter. Then the researchers did this complicated statistical technique, PCOA analysis, which is basically using dimensionality reduction and complex statistics, but they ultimately found that the long chains, which were the vitamin K2 is MK6, vitamin K2 is MK12, and vitamin K2 is MK13, were positively associated with higher MMSE and cognition. So if you remember previously, if you have a higher MMSE, you have less Alzheimer's, and then your cognitive functioning is better. So they found that the individuals that had more long-chained MK6, MK12, and MK13 in their fecal matter was positively associated. They had a higher MMSE. So if the people had more long-chained vitamin K2 present in their feces, then they had a higher MMSE and less chance of Alzheimer's and better cognitive functioning. Now, this is all very interesting, but once again, I would love to see some actual clinical trials seeing that if this actually produces clinically relevant effects and how long you would have to take at what dose. So before I end, remember to please like and subscribe to support me if you like this type of content. Thank you.